the computer. Okay, everybody. So welcome to our April edition of the Sharon STEM Talks. We're super grateful to have Professor Andrew Murray from Harvard joining us. And today is a special occasion because we are celebrating our 4-year anniversary of the Sharon STEM Talks. So to mark the occasion, I'll give a small intro about our series for those of you who aren't very familiar with it. So our program got started back in January 2016 when I approached Mr. Dixon, my former biology teacher at Sharon, and suggested that we get a science program going because we have so many great scientists in our community. Why not bring them together and have them share their work with students? And so Mr. Dixon and Dr. Collins, Ms. Burke all loved the idea, and we got the idea going. And we got our first big break when the ASCB funded us and gave us our first grant through the Compass Outreach Grant Series. Then the Sharon PTSO helped us out as well. And in April, we had our very first inaugural seminar. We had John Lydiard from Biogen give a talk about epigenetics. Since then, the Sharon Education Foundation has funded us also. And for the past 4 years, with support from ASCB and the Sharon Education Foundation, we've been super lucky to bring scientists from all of these wonderful institutions to Sharon to share with our students and our community about the work that they do. And looking at this slide makes me feel very privileged to be living in Massachusetts, to be surrounded by this amazing scientific community right in our backyards. Over the past 4 years, we've heard many talks about biology. We heard Avi Rodal speak about her work on neuroscience and Lowe syndrome. We heard Mikhail Garabedian talk about the cytoskeleton. Lauren Zassadel from Angelica Amon's lab talked about cancer aneuploidy. Joda Georges talks about marine biology. We've heard many talks about physics and astronomy, such as Vonnegut Dogic talking about self-assembly of materials. We talked about gravitational waves. Alan Marsher gave us a talk about how he contributed to discovering the picture of the black hole. Yitzi Kal, a Sharon alum, talks about nanotechnology. We talked about medicine, public health. Heather Goldstone from NPR came and talked about global warming. Dr. Merper gave us several talks about medical imaging. And we had several talks by local scientists who are on the global public health effort in Bangladesh and in Haiti. And part of this series was not only to educate our students, but also to encourage them to pursue careers in science. And so we had several science careers talks as well. Lauren Solano talked about careers in the life sciences. Shauna Buttery from Salt Press talked about scientific publishing. Igor, our class of 2008 graduate, talked about careers in data science. And I've spoken to you guys many times about pursuing your dreams and daring to be wrong. And so after having done this for 4 years, I couldn't think of a better way to celebrate this occasion than to host Professor Andrew Murray for our 4-year anniversary keynote lecture. Andrew Murray is the Herschel Smith Professor of Molecular Genetics and Professor of Molecular and Cellular Biology at Harvard. And he is the director of the John Harvard Distinguished Science Fellows Program. When you've studied in your biology textbooks about cell division and how cyclins drive cell division, you were studying Andrew Murray's work. Throughout his career, Andrew Murray has been studying how the rules of the game work in cell biology, both in cell division as well as in evolution. He is a member of the National Academy of Sciences. Last year, he was president of the American Society for Cell Biology, which has been supporting our Sharon STEM Talks from day one. And today we are super privileged to hear his talk about what sets the speed of evolution. If you have any questions, feel free to unmute yourself or type your question in the chat box. Andrew, thank you so much for joining us. And I will now stop sharing my screen, make you the host, and so you can share yours.
Okay, thank you so much. All right, let me get the screen shared. While I do that, here we go. Oh, sorry, the slides are right at the end. I'm a very disorganized person. All right. Can you see the screen, guys?
Yes, we can see it.
Okay, let me start the slideshow. You would think I've been teaching with PowerPoint, and you would think I would have this down by now. So can you see the correct screen, the one that just says what determines how fast populations evolve and is not the
We see the
presenter view?
presenter view screen.
Okay, I need to swap the displays. Okay. All
That's
right.
correct.
That's better. Okay. So, all right. Um, so I guess what I like to say is how incredibly privileged I am to be here for your 4-year anniversary. I think science education is a super important thing. I'm also sorry, I'm going to try and do one other thing. I'm going to have the participants window up. So just like Vladimir said, you're super welcome to just pipe up and ask questions. And I would really, really encourage you to ask questions. You can also, if you prefer, there's this thing on Zoom where you can put your hand up. And I have the participants up, so if you put your hand up, I will eventually see you, or Vladimir can call out to me. Anyway, I'm super excited to be here. I wondered how Vladimir was connected to Sharon High School, and now I know that he's a graduate, so that's spectacular to give back to your own high school. And it's really wonderful to be here. And like I said, I'm just going to say it one more time. When I start teaching every year, I show a cartoon, which is from a cartoon about a family, and the kid has gone off to college and he's sitting in the front row and he goes, the thing the prof just said was super unclear. I really want to ask a question. But if I put my hand up and I ask a question, I'm really scared I'll sound like a doofus. And in the last panel, it's all the kids in the class with the thought bubble over their head. I'll sound like a doofus. In science, here's the thing. When my kids were little and it was winter, and I was trying to give Barbara, my wife, some time off on Saturday morning, we went a lot of times to two places. We went to the Harvard Natural History Museum, because if you're a Harvard faculty member, you can get in for free, and I'm a cheap bastard. And which my kids now call the Dead Animal Museum. But we also went quite a lot of time to the Science Museum, where we paid up to get a family membership. And there used to be a little section for kids there. And one of the quotes, and there was all sorts of things kids could do. And in the middle, there was a little area, and there were quotes from scientists around it. And there was one from a guy called Leon Lederman, who won the Nobel Prize in physics, and then spent a lot of his time trying to decide how science education should work, and spent a lot of time with the Chicago public school system. And the quote from him is that people who never stop asking silly questions grow up to be scientists. And so if you have questions, please just fire away. Okay? So with that as introduction, what I'm going to tell you about is what determines how fast populations evolve. Oh, and then I also always have trouble with this, which is I have to, there we go. I think I have control of PowerPoint. Woohoo. And so, here I'm going to just, like I said, you would think I know how to use Zoom by now, but it's really true. I know how to use very few things in the world. Okay. So this is a picture of a galaxy. Now, it's possible that in that galaxy there are stars with planets that have life on them, but until recently we didn't think that was that common. And as long as there's nothing alive in this galaxy, this is a beautiful picture. It's a collection of all sorts of stuff. There's wild and crazy physics going on. But except for theological considerations, there's no purpose to any of this. It's just atoms whirling around in space. And so there's a huge difference between non-biological and biological systems. So here, and I'm still trying to get rest control back. Okay. Here's my view of living organisms. Okay, this is the Starship Enterprise. That living organisms are vehicles to disperse genes through space and time. And they are fighting with each other to do that more efficiently because the guys who do it more efficiently persist, and the guys who don't, like the dinosaurs, end up going extinct. And so that means that there's effectively warfare going on between organisms. Everyone's trying to leave more copies of its genes than everyone else. They're trying to spread their genes through space and time. And they keep inventing things in their arms races with other organisms according to the rules of evolution. So we have to show you obligatory quotations. So the first of those quotations, which is a guy from Theodosius Dobzhansky, who is really the founder of experimental population genetics, is nothing in biology makes sense except in the light of evolution. And then there's a less well-known and more modern retort, which is nothing in biology makes sense. Sorry, nothing in evolution makes sense except in the light of biology. And both of those things are true, right? That to understand biology, we have to think about evolution, but to understand evolution, we sort of have to think about the underlying biology and why certain things confer advantages. And so the next thing I'm going to tell you is this would be an easy problem to study if we had the following device, okay? And actually, one of the things you can do in Zoom is if you open the participants box, you can vote and whether you can yes or no. So can I get a vote from you guys about whether you know what this object is? Or maybe in your, can you vote in your version of Zoom or not?
I don't see that option on my screen.
Okay, sorry, then.
We can give you a thumbs up.
Yeah, thumbs up or raise your hand.
Okay, so Zach has his hand up. What's up, Zach?
I was voting. I was electronically voting.
Oh, okay. All right.
In the affirmative.
Okay, what is it?
But Dixon's the one who can really tell us about this. I think this is his favorite topic. Dixon, take it away.
It's a TARDIS, time and relative dimensions in space. Doctor Who.
It's a TARDIS. It's the thing that Doctor Who uses to jet around time and space, forwards and backwards. So if we had this, we don't need me, because we can just go back. We can see how life started. We can see what extinguished the dinosaurs. And unfortunately, Doctor Who only exists inside some BBC studio. And actually, about every 3 or 4 years, Doctor Who undergoes a transmogrification because some new actor gets recruited to play Doctor Who. So we don't have this. So what we have to do instead is, sorry, and I have to keep having to check I'm doing the slides in the right order, is to figure out what's happening. And I'm going to explain what my lab does in terms of this. And it's sort of important to say this. For trying to understand how organisms work now, we have access to the organisms. Like you could experiment on your biology teacher, try and understand how they work, or your chemistry teacher, okay? But in terms of trying to understand how things worked in the past, there are two things you can do. You can study historical evidence like fossils or DNA sequences, or you can study evolution in the lab. But if you study evolution in the lab, which is what we do, you're studying evolution in one place in time, and you're trying to make guesses about what happened in other places in time. So what I claim we do is we do rigorous speculation. So we do experiments and engineering to reveal and test ideas about the trajectories by which organisms evolved. And it's important to say what this is not. It gives ideas and speculation about how evolution happens outside the laboratory, but it is definitely not showing how outside the lab evolution happens, because we're doing evolution inside the lab, and that's different. So I'm just going to give you an evolution refresher. So evolution depends on three things. It depends on inheritance, that children are like their parents. Each member of the Sharon High School class is more genetically like their siblings and their parents than they are to any other member of the class or anyone else in Sharon. So it depends on inheritance. It depends on mutation. So genetic variation is constantly generated. So everyone listening to this talk has about 60 mutations that arose during the cell divisions that made the eggs and sperm in their parents, but most of the cells in their parents' body do not have. Okay? And if you're a high school student and you have children in the future, each one of those children will have about 60 new mutations. You'll give them 30 and your partner will give them 30. And it depends on selection. This is the whole Darwinian principle that some genotypes get to leave more progeny than others, and that changes the structure of the gene pool. So my contention is that we have trouble understanding evolution for two reasons. The first is it's dominated by a succession of very, very rare events. Things happen that you have to wait a long time for, that are extremely unlikely, but they give advantages to organisms, and therefore they get propagated. And the second thing is, amongst all those unlikely events, if some of them are a little bit less unlikely than others, those are the ones that are likely to lead to evolutionary change. So it's the relative probability of different unlikely events that determines evolutionary paths. And the last thing is, as I said before, we can look into the past for evidence about evolution. So the classic one is fossils. So I guess what I'm going to do is, if you wanted to be, because you can put your hands up, I think, right? If you wanted to be a paleontologist when you were a kid, put your hand up. Okay. So I guess you're an unusual group of people, because usually if you talk a bunch of high school kids who are interested in science, at least someone wanted to be a paleontologist and study dinosaurs when they were little. But not everything gets preserved from dinosaurs. And the other sort of evidence is we compare DNA sequence and we try and infer what DNA sequences looked in the past. And all that evidence is imperfect. So I'm going to talk about three sorts of evolutionary change. So one is continuous gradual change. And one example of that is influenza. Okay. So lots of you got flu shots last year. Actually, this year, probably remarkably fewer people got flu than normal, because we're all social distancing and flu spreads by social contact. But you need a new flu vaccine every year, because flu changes so that it can evade the antibodies that the sort of global human population made last year, because those viruses can't infect, again, people who got flu, but slightly different viruses where the structure of the key proteins that they use to bind and get ingested by human cells, where those proteins have changed, they can infect you. And there's some aspects of the domestication of plants and animals that are probably the same. The experimental version of this is to ask cells to grow faster in what I'll call standard environments, the environments that scientists use to grow cells in the lab. So that's one mode. A second mode is the appearance of novel traits. And one of the big questions in the world is, what does what is evolutionary novelty? It turns out to be remarkably hard to define, but an unpleasant form of evolutionary novelty in people is cancer. They're examples of what biologists call adaptive radiation. I'll give you one in a second. And some of the changes that have occurred as plants and animals are domesticated are pretty radical changes. And so here's an experimental example. The organism that I'm going to talk to you about today, the bakers and brewers yeast, Saccharomyces cerevisiae, is in the hands of laboratory scientists, a single-celled organism. So in my lab, we've evolved it to be multicellular. So we'd claim that's novel. And we've evolved it to have a circadian oscillator, which is the sort of thing that's inside your biological clock. You have a circadian oscillator that tells you when to get up. If you're a teenager, it probably tells you to get up at 10 in the morning, and your parents have to be there with a pitchfork to get you up at 7 in time to get to school. And that oscillator is responsible for jet lag, because when you fly to somewhere like Europe or the Far East, your body thinks it's one time when the sun thinks it's another. And the last example, which I'm not going to talk in any detail about, is what we call evolutionary repair. And so what happens is, if you look at various aspects of the biology of cells, you find things that biologists have confidently asserted are universal. Everyone has this thing. And then you look at enough different organisms, and you find, to your shock and perhaps a little bit to your horror, that things that people claimed were universal are missing in certain organisms. And there's a laboratory version of this, which is to take an organism, remove some gene that cripples but doesn't kill it, and then ask if you can accumulate mutations, which is what evolution is, and persuade the organism to recover from removing this important thing. Okay, so now I'm going to tell you what evolutionary novelty looks like, and I obviously can't spell evolutionary. And so this is an example about what happened in Hawaii. So Hawaii is about 2,000 miles over the sea from North and South America. And in Hawaii, the Hawaiian Islands arise because there's a hot spot in the Earth's mantle. And as the Pacific plates pass over it, about every million years, the lava bursts through and it makes an undersea volcano. And eventually that volcano grows so high that it becomes an island. And so there's the Hawaiian island chain. The island closest to us, the big island, is the newest. It's about 1 million years old. And as the islands get smaller, that's because they're older and older, and the volcanoes have gone extinct, and they're gradually being worn away by erosion. And so, because Hawaii is so far away, the winds and the currents blow from the Americas to Hawaii. A very small fraction of all living things have made it to Hawaii. It's like a very cut price airline with very crappy planes, and most people don't make it there, okay? And so here's an example. The thing that is shown at the top of the slide is the Californian tarweed. And there are lots of plants like this in the continental Americas. They're spindly plants, they have little yellow flowers, and they all look pretty much like each other. One of those plants got to Hawaii about 5 years ago. 5 million years ago. And it has given rise to the crazy diversity of things that are shown here. So the thing on the left is what's called a cushion plant, because it sort of looks like a cushion you could sit down on. The thing in the middle is a tree. And the thing on the right is this weird spiky plant. These guys are more related to each other than the tarweeds in the continental Americas are related to each other, but they look a lot more different. And the idea is that when they got to Hawaii, because most plants didn't make it there, there were all sorts of ecological niches that these guys could inhabit if they mutated and accumulated mutations and evolved crazy fast. Okay, so that's a real world example of evolutionary novelty. And here's a laboratory example of gradual evolution. So this is how people study evolution in the laboratory. So this is a picture of a test tube. There are a bunch of cells in there. They could be yeast, they could be bacteria. Their ancestral genotype is colored orange here. And you grow them in some particular condition. And you hope that mutants arise that will enable them to grow faster. And so the mutant cell here is shown in yellow. And after the cells have grown, and they've used all the sugar, for example, in the test tube, you dilute them into fresh media. Now, for things to work, your fabulous yellow cell has to survive that dilution, and quite often it won't. But if it does, and it reproduces faster, its frequency in the population will increase. And so here are two examples of this. So there's a guy at Michigan State University, and he's been studying how E. coli, the bacterium that you can find in your intestines, adapts to growing on a much lower glucose concentration. It's about it's a 200-fold lower glucose concentration than people normally use. And he's been doing this experiment now for 30 years. So the cells have gone through 70,000 generations. And the cells keep evolving to grow a little bit faster under these particular conditions. Humans do the same thing. So people who breed crops keep breeding things like corn to each other and selecting for the corn plants that give the highest yield, and they keep repeating that. And people have been doing that for over 100 years. And it's still true that by this selective breeding, they're able to keep a little bit each 5-year period, increasing the agricultural yield of corn. Okay. And so one of the questions that you could be asking yourself right now is, why am I listening to this guy? He's going to talk about experiments in evolution, but that's not real evolution, because real evolution happens outside the laboratory. And so I'm going to give you a list now of the reasons we do this, the advantages that we have from studying evolution inside the laboratory. And the first one of those is we know where we're starting. When you study real evolution, most of the time you don't know what the starting point was, what the organisms looked like, what their genomes looked like. We know what selective pressure we're applying to the organisms. We can keep a living fossil record. So yeast, it's easy to freeze them down. And when you're interested in asking what happened in your experiment, you can thaw them and get them to grow again. So we can keep a fossil record. Really importantly for us, but I'm not going to talk about details, the ancestral and the evolved forms can interbreed, right? So there's a lot of excitement about the difference between humans and chimpanzees. Why are we so different from chimpanzees? Now, in some nasty science fiction movie with evil, unethical scientists, one way you could figure out that question, in principle, is if you could breed humans and chimps together, you could make lots of human-chimp hybrids, and you could ask which ones look more human and which ones look more chimp, and you could analyze their DNA and try and find the genetic mutations that gave rise to all the things that make us think we're so damn special. Okay? So there's good news. Not only is this experiment ethically horrific, but it doesn't work. You can't actually interbreed humans and chimpanzees. But with the experiments we do, we can interbreed our starting populations and the evolved ones, and we can use that to track down mutations. And the last advantage is that you can do multiple parallel experiments. So Stephen Jay Gould, who was a famous scientist who wrote about evolution, some of you may have read books of his, he talked about whether you could replay the tape of evolution and whether things would look different. At least in the laboratory, we can do a whole series of experiments at the same time, and we can ask how similar are their results. So sadly, we have disadvantages as well as advantages. One of them is that time, our time scale is set by how long it takes a graduate student to get a PhD or a postdoc to do a postdoctoral fellowship. And typically that's about 5 years. So with the exception of some experiments like Rich Lenski's experiment, which has propelled the careers of lots of graduate students, and it's lasted longer than any of their individual PhDs. Time's limited, and how big you can make populations limited. You can't fill Boston Harbor with yeast. And the second thing is that the environments in nature are much more complex than the ones in the laboratory. There are lots of different species competing with each other. There are seasons in the year. Environments are spatially differentiated. And as a result of those differences, we don't yet know how much what we learn from laboratory experiments is applicable to things in the real world. I think there's some strong arguments, and if people want to discuss it, as to why what we learn in the lab is useful for studying the real world, but at the moment they're just arguments. And I guess one of the things I should do right now is stop and see if anyone wants to ask questions.
Yeah, Andrew, I have a question for you. So
Yes.
what if you made a unicellular yeast and a multicellular yeast? What's the progeny, if that's possible?
So what's really interesting is we've done that experiment. So John Koshmanez, who did the experiment, has to know the answer. And it is super embarrassing that I don't know what the answer is. I have a prediction of what the answer was, but I need to say this carefully because I don't know the answer. The answer is that when you looked at the cross between the multicellular strain and its unicellular ancestor, they were unicellular. And here's why I'm speculating that rather than knowing. Because in that, what we discovered is that almost all the mutations in almost all the experiments that we've done that produce evolutionary change are what a geneticist would call recessive mutants. They're mutations that make proteins work worse or not at all. And if that's true, that if you cross them to the ancestors who have working versions of all those genes, the phenotype should look much more like the ancestor. I will, it's a great question. I will email John and see if I can find out what the answer is.
Thanks so much.
Oh, you're so welcome. Any other questions that people have? Oh, okay. And so I already told you that we do this with our friend, the budding yeast. It's a very good friend of mine. I'm fond of beer. I'm fond of bread. But the scientific reasons are it divides really quickly. It has a doubling time of about 90 minutes. This is an electron micrograph of a bunch of yeast cells. They're roughly 5 microns in diameter. You can get it to proliferate sexually or asexually. And that's actually a huge advantage to us in tracking down mutations. It has really good genetics. As haploids, as diploids, we understand how sex works. Of all the eukaryotes, it has the best genome-based tools to find mutations, and we know more about the biology of these cells than we do about any other sort of cells. So one of the things that I have to tell you about before I tell you, I'm going to tell you about one experiment. If you're sitting there like, is he going to tell us about an experiment? I'm just waiting and waiting and waiting. We're going to get there. We're going to get there fairly soon. Okay? But we have to give you a sort of prelude. So we're going to evolve cells, and we're going to declare that they have evolved to be better, or in evolutionary geneticists' words, fitter, if they can outcompete their ancestors. And we need a way to measure whether they're fitter. So the way we do that is shown here. We take some evolved cells, and they're colorless, and we take a version of our ancestor and we label it, we genetically modify it so it expresses something called yellow fluorescent protein. So this is a protein that's ultimately modified from proteins that live in jellyfish. And when you shine blue light on it, it shines yellow light back on you. So you mix those guys together, you grow them under the condition where you did the evolution. And when you finished, you measure the ratio of the colorless cells and the yellow cells. And the machine you use to do that is called a fluorescence activated cell sorter. So that's what FACS stands for. And if you do it a bunch of times, you grow the cells, you measure the ratio, you dilute them, you measure the ratio, you can plot the log of the ratio of the evolved strain to the ancestor. And if you plot that against time and you express time in generations, the slope of that red line is how much fitter your evolved cells are than your ancestor. So that's how we actually measure this thing called fitness. Okay? It says slope equals fitness. Okay? So the whole topic of this talk is what sets the speed limit for evolution. And our goal is what we'd like to have is a quantitative understanding of evolution. Okay, so here's a mathematical model. So this mathematical model is the rate of change of fitness. So the abbreviation that population geneticists use for fitness is W. So dW dt just means the rate of change. And F means is some function. I don't know what the function is, but it's some function of all these things in the bracket. So on another slide, I'm going to tell you what they all are. I'm going to tell you quickly now. NE is a measure of the population size. UB is the beneficial mutation rate. UD is the rate at which deleterious mutations appear. That Greek letter rho of S is something about the distribution of how big or how small the beneficial or deleterious effects of the mutations are. There's something called epistasis, which I'll explain you what that is, and there's sex. Woohoo. The problem is that this is hard to do because most of these parameters, all the ones listed here, we don't actually know what their quantitative values are. And they also have a range of values for different mutations. We just don't know what they are. And the second problem is, even if we did, the math is super hard. Okay? So what we're going to do is do what scientists do all the time, is they make the problem really, really simple. Okay? So here's a way, and this word, which is reductionism, gets sort of misused quite a lot. So there's a lot of talk now about holistic approaches to science, like science was some form of aromatherapy. And that it's bad to be reductionist. It's bad to think of, biology all being to do with the chemical and physical interactions of molecules, which, trust me, is all biology is, but that you could be studying the problem in too sort of minute a way. That's not what I interpret reductionism as meaning. I interpret reductionism as meaning you should find the simplest possible example of a problem because that's going to be hard enough to study. And I'm going to give you an analogy. Imagine you arrived from Mars and you want to understand how cars work. So you could come to me and tell me that, I could go to Mars and return in their spacecraft if they could borrow my car. So our car is a Honda minivan. It's like 3 years old. It has all sorts of complicated things on it. Like I can't start the car unless I have my foot on the damn brake, right? It has, electric mirrors and stuff like that. And so if the Martians just start fiddling with stuff, they're really going to have trouble. They're like, why does someone have to push on this pedal to make the car go, even though under other circumstances, pushing on this pedal makes the car stop? They would be much better off making the swapping vehicles deal with someone who drove a drag racer, right? Because a drag racer has a big ass engine, a gearbox, and some big ass wheels at the back to make it go really fast. It does not have any video screens to pacify the children in the back. It doesn't have any electric mirrors. You don't have to put your foot on the brake to start it. And the drag racer is the reduced form of an automobile, okay? So here's the principle of reductionism. You find something interesting you want to study. The Martians want to know how cars work. You define your terms. You say what it is you want to study about it, right? The Martians might just be interested in how do you make the damn drag racer go so fast and not really be worried about how you stop it. You state your question in the most general possible form. You find the simplest possible example of the problem. That's a drag racer. You define the level of what I call the level of abstraction in which you want to study the problem, right? Because maybe for the drag racer, you don't really need to know how the rubber of the tires is made. You're just going to assume that you can make something that grips to tarmac, and that will be good enough. And then you watch, you think you know something about how the drag racer works. So like you decide, I'm going to remove the gas pedal. Does it take off when the light turns green? If it doesn't, that suggests a hypothesis, and you make predictions. To test the predictions, you do more observations, and you go round and round. And then the final thing is you try and extrapolate the understanding from the specific example to the general problem. So actually, I have a question. So we have some teachers here. Have the kids here heard about the lac operon by any chance?
The ones in AP Biology and Biotechnology, some of which are on this call.
Okay. So some of you have heard about these two crazy French dudes who wanted to know how E. coli switched from eating glucose to eating lactose, which is the sugar in milk. And their names are Jacques Monod and Francois Jacob. They're super wonderful scientists, and they were heroes of the French left during World War II. And they figured out all this stuff. And afterwards, Monod wrote about it, and he said, what is true for E. coli is true for the elephant. Okay? Now, you can have a big argument for a long time with people who specialize on understanding gene regulation in elephants or humans about whether it's really true that gene regulation in mammals looks essentially identical to the way it does in E. coli. And they'd argue about it, and people would fight, and they'd get a little tipsy as they were arguing. What you can't argue about is the fact that the ideas that Jacob and Monod put forward told people how to study gene regulation in mammals. And even if the details are important in critical ways, the fact that someone had a model for how to do the studies and how the problem worked made it possible to work out things in mammals which are much more complicated. So that's what I mean by extrapolate from the specific example to the general problem. Okay? So here are those parameters that were in that mathematical model. So there's the reproductive fitness of an individual. There's also the mean reproductive fitness of a population. there's this thing called the effective population size. In most populations, they go up and down over time. That drives mathematicians crazy. So they want the little red line on this diagram, which is a population whose size is constant, who behaves the same way as that crazy sawtooth population, which is going up and down. And that's something they call the effective population size. And you'll notice on this graph that that size is much smaller, it's much closer to the bottleneck size of the populations than the peaks, okay? Then I told you there's the beneficial mutation rate, there's a deleterious mutation rate, there's how good or bad the mutations are. So this graph shows the frequency of different sorts of mutations. And S is how much better, these are beneficial mutations, so S is positive, how much harder it is to get ones that help you a lot. Okay? And then epistasis is this interaction amongst mutations. So S12 means the fitness effect of having both mutation 1 and mutation 2. They each have their own fitness effects, S1 and S2. And delta is some number that shows the sign, that's whether it's positive or negative, and the magnitude of the epistasis. So this is a way of saying sometimes if you make 2 mutations, they're way better than you thought they would be based on their individual effect, or they're way worse. And last of all, there's sex, like how often, especially in something like yeast, which doesn't have to have sex in every generation, how often do you have sex, whether you're inbreeding or outbreeding? So this is all complicated. So there are 3 approaches to solving this problem. One is to do experiments. One is to do theory. So this is a guy called Michael Desai, who was a graduate student who worked on this problem. And his advisor was a theoretical physicist called Daniel Fisher. And the last thing you can do is simulations, but for the sake of time, I'm not going to talk about simulations, okay? So what Michael and Daniel did is they went off and they were like, okay, let's see how simple we can make this problem. Let's make crazy simplifications and see what happens. So the first assumption was that the population size is going to be constant, that all the advantageous mutations are going to have exactly the same benefit, which we're going to call S. There are no deleterious mutations. This is a world that just gets better. The mutations interact with each other additively. That is, if you have 2 mutations, their effect on the fitness is twice S, because each individual has an effect S. If you have 3, it's 3 times S, and so on. And this is the British version of evolution, so there's going to be no sex. Okay? So now, there are 2 regimes for how populations can evolve. One is, and in this diagram here, going from top to bottom, how wide a color is how many individuals there are. And the x-axis is time. And so what you see is the population starts out, I guess we'll call that color magenta. And a mutation arises, and that mutation is a little bit fitter, and it's going to spread through the population. And as it spreads, it will eventually take over the population. And now you have to wait a while before the next mutation that's going to make it better still arises and spreads. And so now if you draw underneath that a diagram that represents the fitness of the population, it goes up each time a mutation is spreading through the population. It sits on this plateau for a while. The next mutation spreads, and so on and so forth. And you don't have to know very much about evolution, and you can calculate the rate of evolution. It's S squared, where S is the effect of an individual beneficial mutation in this crazy world where all the beneficial mutations have the same advantage, times the effective population size times the beneficial mutation rate. But things get complicated if the population is bigger, because now the first mutation occurs. It has to spread through a bigger population. And before it gets done, the second mutation happens on top, and then the third, and so on and so forth. So now if we plot how the fitness changes with time, it's much smoother, and the math is hard. So what Michael decided when he was a graduate student was to figure out the math. And to do that, you consider a newly arisen mutation, the benefit it causes to the fitness is bigger than 0, but much less than 1. And then there are 2 regimes where these mutants behave. So when you have lots of the mutants, when the number of mutant cells is much bigger than 1 over S. So you can imagine that S is 1%. So that's 0.01. So 1 over S would mean that you need 100 mutant cells to be in this regime. The number of mutant cells behave what a physicist calls quasi-deterministically. It's like you can write down an equation. You don't have to worry about chance fluctuations. The fraction of the mutant cells increases exponentially with time. For a given value of their benefit, the time to fix it takes the mutant to fix, that is spread all the way through the population, is proportional to the log of the population size. And for a given population size, the time the mutant takes to fix is inversely proportional to how much fitter it makes the population. But you also have to deal with what happens when there are not very many of the mutant cells, right? When they're less than 1 over S, less than 100 in our case. Then what happens to the mutant cells, it's sort of like tossing dice, whether you get more or less in the next generation. And only a small fraction of the mutations that occur have the number of mutant cells get high enough to survive genetic drift. So if you don't get to 100, you're likely to get hosed out just by chance fluctuations. And different mutants take different times to reach this size that guarantees their survival. So there's 2 sorts of math. There's math which is just smooth functions, and there's math where you have to worry about chance effects. And what you had to do is to stitch those things together. And the last thing I would have to say about when the things are few is the interval between the survival of successive mutations varies. So you have to take that into account too, okay? But now I'm gonna try and show you this all on a graph, okay? So here is a diagram of our population. It started at fitness 1. A beneficial mutation arose. So its fitness is 1 plus S. And it's survived, it's gotten to be more than 100 cells. So it's fitter. So its abundance is going to go up. That's the green arrow. And the cells, the wild type cells which produce immune are less fit, so their abundance is going to go down. Okay. Now, if the population's big enough, as the guys who have fitness 1 plus S increase in size, there will be enough of them so that they'll produce new mutants. They all have the same fitness effect, so their fitness will be 1 plus 2S, right? Those guys are much fitter than the mean of the population, so they'll increase fastest. The guys whose fitness is 1 plus S are still fitter than the mean of the population because there's still a lot of the ancestors left, so they're going up, but more slowly. And the ancestors who are the least fit guys are less fit than the mean of the population, so their abundance is going down. And if you keep waiting, what will happen is you'll get a picture like this. So now, the guys in the middle, because this diagram is exactly symmetric, the guys in the middle are exactly at the mean fitness, which is now 1 plus 2S. They're not going up or down. There's still a few ancestors left. They're much less fit than the mean, so their abundance goes down quickly. There's 1 plus S, they're a little less fit than the mean. Their abundance is going down. And the most fit guys are the guys who have 2 extra beneficial mutations relative to the mean. They're at 1 plus 4S, and they're going up the fastest. If you think about this for a second, this whole distribution is just going to march to the left. As long as you don't run out of beneficial mutations, the guys at the nose, the 1+4s guys, will eventually get numerous enough to give rise to new beneficial mutations. So they'll hop one over to 1+5s. And eventually the ancestor, the tails, will get driven extinct. So now selection is driving the guys at the tail extinct, and mutation is producing new guys to drive the nose forward. And so if you look at the graph a little while later, it's just marched across, okay? And if you think hard enough about it, if the tail stretches too far back, it will be much less fit than the mean population, and it will get squeezed out faster. So if the tail drags a little, it should speed up. And if the nose has a little trouble going forward, you should keep building up the fittest class, and it should jump forward. So at least in a sort of time averaged way, the nose and the tail have to go at the same speed, and the distribution has to stay the same. So you get out your pencil and paper, and you solve two equations, one for the speed of the nose and one for the speed of the tail. I just told you that over long times, the nose and the tail have to go at the same rate, because if they don't, the distribution will get fatter or thinner. So you have to make those two equations equal to each other. And when you do all that, you get a scary, complicated equation that looks like this. So this is, again, the rate of change of the fitness, dW, dt. And then S is the fitness effect of the beneficial mutations, N is the population size, and UB is the beneficial mutation rate. And what all that stuff means is the speed is proportional, roughly, to the logarithm of the effective population size, and it increases with the logarithm of the beneficial mutation rate. Okay, so great. You're sitting here, it's 8:04, and you're like, thank you, dude, nice theory. Does this theory explain anything? So what Michael did at this stage was to do an experiment. So he evolved cells, so I'm just showing you the diagram of evolving and diluting again, to grow faster on low glucose. So he took rich medium and he reduced the glucose concentration by a factor of 40. He kept the cells growing exponentially by diluting them every 9, 15, or 21 generations. He used, and those different dilution times give rise to different effective population sizes, which vary by a factor of about 2,000. And he also changed the mutation rate. So the wild type rate at which yeast cells make mistakes is once in every they make a mistake every 2 billion base pairs per cell division. And if you make mutators, which are called MSH2, and this is a mutation that gives rise to human cancer in the equivalent human gene, they make mutations about 10 times faster. So now we're going to actually do an experiment, and we're going to compare theory and experiment. So here's the graph, okay? And so there's a whole bunch of things on this graph. The x-axis is the relative value of the logarithm of the product of the effective population size times the beneficial mutation rate. And the y-axis is the average speed of adaptation in fractions of a % per generation. Okay? And so now I'm going to show you a bunch of lines. So this blue line is what it would look like in the 1-by-1 theory, where each mutation occurs, spreads through the population. You have to wait for the next one. Okay? So it would show this crazy curve, and it would get faster and faster in a wildly nonlinear way as you increased the logarithm of the effective population size times the beneficial mutation rate. So here are cells with the wild type mutation rate. So there are two sorts of points here. The open point are the points for Michael's theory. The closed points are for 6 independent replicate experiments, okay? Now, when people show you the data for replicate experiments like this in biology, usually some of the difference between the points is they're just not very good at measuring things. It's what you call a measurement error. But in this case, because these are populations evolving and every population doesn't evolve exactly the same, some of the spread between these points is probably real biological variation. So these are the guys who have the wild type mutation rate. And here are the guys who are the mutators, who have a mutation rate which is 10 times higher. So the theory is the open triangles and the experiments are the closed ones. You will see that there's much more variation here for the mutators than there is for the non-mutators. But again, if you take the average of the mutators, it's a surprisingly good fit to the theory. Okay, and so here's the last slide that I'm going to show you. And this is the WTF slide, okay? I'm just going to go back. We made a bunch of really crazy assumptions here, which we know are not true, okay? That the mutations interact purely additively, that they all have the same effect. So we made crazy assumptions, and yet the data fits the model that comes from the crazy assumptions surprisingly well. And when that happens, scientists are forced to go backwards and go like, huh? How can it be that I made crazy assumptions, but it still sort of works? And so here is the answer to that question. I just said, you can measure this in experiments. All mutations do not confer exactly the same benefit. There are lots of things that confer little tiny benefits, and there are many fewer that confer big benefits, okay? But here's what happens. There's some special size of the effect of the mutations, which we're going to call SF for S effective. And the things that cause fitness increases that are much smaller than this, and there are lots of them, are eliminated by what's called clonal interference. And what clonal interference means is in that distribution that was who's fitter and who's less fit, the guys at the back of the distribution at the tail, they're the living dead. They're the zombies. They're almost certain never to contribute to succeeding populations because they're so much less fit than the guys at the nose. And most of those tiny effect mutations occur in the living dead. And if you occur in the living dead, it doesn't matter whether you occurred or not. There are also some mutants that cause big effects, even bigger than this S effective size. When they occur, they are likely to increase the fitness of the population because they can jump things in the tail up to the nose, but they just happen so rarely that they don't matter enough. And so what that means is that evolution is dominated by a narrow range of fitness effects. So there is effectively a speed limit. We can compute the speed limit. And there's always a caveat. Scientists always finish with a caveat. The caveat is valid as long as the distribution of those beneficial effects falls off exponentially, that it's exponentially harder to get a mutation that has big fitness effects than it has small fitness effects. So that's a story about the speed limit for evolution and how you can calculate it. And I will be happy to take any questions at all that people have. And to do that, I'm going to turn off, I'm going to stop sharing the slides. Great. I'm going to go to the gallery. And I'm especially interested in answering questions that students have.
Thank you very much, Andrew. So if anybody has a question, unmute yourself and ask away. Okay, I guess I'll get started in the meantime while people think of questions.
Sure.
So have you noticed that the mutations that increase the fitness happen in a particular type of gene that does a particular function?
So what's crazy about these experiments is we don't know in these particular experiments what the mutations are.
Okay.
Right? So there are lots of experiments we've done since where we do know what the mutations are. And for some of the experiments, we not only know which genes got mutated, but we understand why those mutations help the cells get fitter. And in this particular case, so I guess what I would say is I got inspired to do experiments on evolution, which is not what I ever thought I was going to do, when the genome of yeast got sequenced and it was clear that you would be able to track down mutations. And so early on, we had a way, this happens a lot in science. This is sort of like your thing about, you know, making mistakes and the mistakes help you. This is sort of the opposite, making mistakes and or thinking that you can make things work and it isn't really true. So we developed a way that we thought would enable us to track down the mutations. And then we did what scientists often do is we did an experiment where we knew what the mutations were and asked if we could find them, mutations that had very strong effects. So we did that experiment and we found the mutations and we published a paper and we ran around and celebrated. And then we tried to use that method on actual experiments where we had evolved interesting traits, and the method failed really miserably. And so we didn't get to a stage where we could figure things out until what's called next generation sequencing came along, and you just sequence the DNA and it works like 8 million. That's exactly the same basic method. The method that we published, I'm so happy with, now actually works. And roughly speaking, we still have the DNA in a freezer from Michael's experiment, and we could go back and try and figure out what the mutations are. And we've just been, I mean, this happens all the time in science. There's stuff you could go back and do, but it's easier, or people who work in labs are much more excited about doing really new things than going back and sort of fixing up or figuring out what went on in old experiments. So we just don't, for this experiment, we just don't know where the mutations are.
For sure. Thank you. Another question?
Sure, I'll ask a question. Can you hear me?
Yeah, you can show your face too if you want.
Moving around,
Hey.
kids in the house, trying to shield everyone from their noise. All right, so I had a question about the equation of, I guess the set of equations. Just wondering how applicable and accurate you think it is for sexual reproducers.
What do I think about that? The exact sort of complicated equation I put up there won't be the right equation because you mix up genotypes. And so there's a big, huge debate in evolution about what is sex good for, right? So, you know, what I got taught in high school and then in college is like, evolution's so great, it, you know, puts together combinations of genes that are more beneficial, right? By recombination and independent assortment of chromosomes. Unfortunately, if you have things that work really well together, and now you do recombination, you can bust them and break them up just as easily as you can put them together. And so in principle, you look at it at first sight, it's like, well, this is sort of swings and roundabouts. What you gain on one and you lose on the other. And so what people, so Michael Desai, who was a graduate student then, now works down the hall from me. And so they did experiments to show that in yeast, sex really does make evolution faster. And the principal thing it does, so remember I was talking about how if you're in the tail of the distribution, you can have good mutations, but it's really hard for that to contribute to evolution because that good mutation has to sort of leapfrog you up to the nose. So one way of thinking about that is that that's a good mutation in a bad genetic neighborhood, right? That the mutation would increase the fitness, but it's in a genome with a lot of crappy mutations, and so it just can't help enough. So what sex does by shuffling up the mutations is it can rescue good mutations from bad genetic neighborhoods. And so it increases the number of the mutations that have occurred that can push the nose forward. And so both in theory and in experiment, it turns out that that's true. Does that make sense?
Yeah, some sense.
Okay.
Thank you.
You're very welcome.
I guess for the seniors on the call who are heading off to college soon, what advice do you have for them when they go to college?
My advice, if you're about to go off to college, is have fun. And the biggest thing is, like, I advise freshmen at Harvard, I advise 3 freshmen a year. And some of them come into my office. I'm like, what are you thinking of doing? They're like, I don't know. I just got to college. I sort of want to check it out. And there are other people who walk in, they open a little folder and they pull out a color-coded plan of exactly which class they're going to take over all 4 years. And they're like, I need you to look through my 4-year plan and tell me that this will get me to medical school or titan of industry or whatever it is. And my response to them is, the best thing to do would be to light a match and burn this piece of paper, because you can't walk in with that plan. You should try stuff like, one of the scientists I've mentored is a woman called Rachel Dutton, and she decided, there's a lot of excitement these days about microbial communities, you know, collections of bacteria and fungi that do stuff together. And there's this big thing about the human microbiome. You have these thousands of different species of bacteria in your intestines, and they contribute to health and disease, and it's sort of a big deal. Super hard to deal with them because they're hard to culture in the lab. And so what Rachel was, her attitude was, I want to study microbial communities. I want a community that's simple, it's reproducible. I hope this doesn't put anyone off cheese, which is one of my favorite foods, but the rind on cheese, unless it's Kraft, is a collection of fungi and bacteria, right? And so the people who make cheese obviously have figured out, A, to make sure that this doesn't kill people, and B, to make it reproducible, because, you know, like if you sell Stilton, it's supposed to taste like Stilton, not cheddar, and vice versa. And so Rachel decided that the thing to do was to use this as a model microbial community. And she's made fantastic progress on this, right? So why am I telling you this? Rachel grew up in San Francisco, or outside San Francisco. And she went to college. She wanted to be the weather person on TV. So she was majoring in communications and stuff. And there was some irritating, boring, stupid distribution requirement where she had to take a science class. And she took a biology class, and she suddenly realized, oh, this is why I'm on the planet. I'm on the planet to be a biologist, right? You know, and then she worked for a couple of years as a technician before she applied to graduate school. And now she's back at the institution she was an undergraduate, UC San Diego, as a professor. And so to me, that's and I know actually a surprising number of scientists where that's true often, that you know, they were sort of milling their way through a college career, and suddenly they went to a science class and they were like, holy shit, this is what I'm supposed to be here for. And, you know, and I know people who've come to Harvard, you know, the people I see as freshman advisees are typically scientists. And there's a substantial number of those who have suddenly realized, no, I'm interested in public policy or stuff like that. So I guess, okay, so what's my advice? Go with the openest possible mind. Ask questions. Be the kid who puts your hand up in class and goes like, I have a question. Either I think this means this, or I don't understand that. Every time you put your hand up in class and ask a question, you will be helping yourself, and you will be helping at least 6 other people who have the question who are too chicken shit to put their hand up. So, and work hard, play hard. You know, I mean, you know, it's fun to be, you know, I mean, here's the thing about scientists. You know, why do we do this crazy stuff we do? Well, we sort of can't help ourselves. It's like we are more or less compelled to do this stuff because it drives us crazy not knowing the answer. But we still need to take time off, and, you know, whether it's playing the violin or watching the Red Sox, like, you know, the Nesson are replaying the Red Sox season in 2004 tonight, tomorrow night, and the night after, the first 3 games against the Yankees, which they lost. I'm not going to watch those ones. I don't need that pain again. But I'm going to watch the 4 nights after that. And so it's, you know, live a somewhat balanced life.